Hello and welcome to this video on Agar, the basic gel of researchers and gelling agent of choice for vegans and vegetarians. Agar is a solid gel that very much resembles jelly. Unlike jelly, which is made from animal collagen, agar is made from plant-derived polysaccharides. Agar may also be known as China glass, China grass, China isinglass, Japanese canton, and Japanese gelatin. Agar itself isn't a single product, rather it's made up of two different products that, when they come together, form a polysaccharide, these being agarose and agaropectin. Agarose makes up the vast majority at about 70%, although this will vary slightly from species to species you derive it from. Agaropectin makes up the remaining 30%. The agarose itself is made up of a repeating series of units. These are linear polymers made up of agarobioes. The agarobioes itself is made up of a disaccharide, or two molecules kept together, these being D-galactose and a rather complicated galactopyranose. The agaropectin is a jumbled mix of small molecules. It's otherwise described as a heterogeneous mixture. Heterogeneous just being a fancy way of saying a mixture of different things. For the most part, it's made up of alternating units of D-galactose and L-galactose, with a modified side group. Generally speaking, this will be sulfates and similar, although there are some pyruvates and glucuronidates. In order to get agar in the first place, you need seaweed. However, not just any seaweed will work. The most common species is going to be ogonori, or red seaweed, but by no means is it the only option, just one of the most common. Agar itself has a number of applications. Perhaps most novel would be as a laxative, and also as an appetite suppressant, but more frequently you'll find it as a vegan substitute for gelatin, where it is used as a thickener in all sorts of products. This owes to the fact that, much like gelatin, it will form a semi-solid mass, which, if you emulsify it properly, will cause a more consistent, almost custard-like consistency. You may also find it used in brewing as a clarifying agent. We've repeatedly compared agarose to jelly or gelatin for a very simple reason. The two are very similar. The primary distinction between the two is where they're sourced from. Gelatin is pretty much produced from the collagen of livestock. It's generally speaking always going to be from animals. This would be the bones, skin, cartilaginous tissues, and tendons. But agar is not. Agar is from the plant cell wall for the most part, and so this is why vegans and vegetarians are fond of it, by comparison at least. The other major difference is the temperatures at which they operate. Agar, or as it is properly called agar, is made using boiling water, while gelatin can be made using just warm water. The reason why you need boiling water for agar is that the agar molecules need to be broken down in that temperature so that they can reform. As a very broad rule, but mostly consistent across different kinds of agar, you find that it melts at about 85 degrees Celsius. It begins to form a firm gel at about 65 degrees Celsius, and you could consider it a solid effectively at between 30 and 40 degrees Celsius, although the exact temperature will vary slightly based on the concentration of agar being used. Unlike jelly or gelatin, agar is, generally speaking, far more resistant to shear forces, and this is one reason why you can use it as a ballistic or similar gel, but basically to test it in lieu of having an actual material available that properly plays the place of tissues. As mentioned, agar can set at a relatively low temperature, and more so at room temperature, where it does so more quickly. And gelatin can do the same, but it takes far longer, and generally performs better when put into refrigeration. Depending on the recipe for which you are using agar, there will be considerable variance, although some may argue that it's more subtle. That depends on how you view the mouthfeel. The biggest issue is going to be how firm, or not so firm, the dishes are. If you use gelatin, the dishes would be considerably less firm than with agar. Generally speaking, agar will always be firm, even when you put it back into a warm environment. Although it can be redissolved, if you were to boil it again, it's far more difficult, whereas to an extent, jelly can be. Generally speaking, agar is used in concentrations of between 1 and 2% by weight, although some are used at a higher concentration, and some are used for far lower concentrations. For example, some research into motility can use as little as 0.5% or even lower again. The advantages of agar over other options like gelatin particularly are that it is completely odorless and tasteless. This means that it can be very useful if you're trying to create products that aren't relying on the flavour. So for example, with cosmetics, where you don't want any other odours, tastes or similar attached to the product, particularly if the individual does not want to taste like a side of meat. At the same time, it's also far more sustainable, let's say. After all, it is seaweed, and it's far easier to farm than trying to get products from animals that are often in competition. But even for food products, it's the same thing. Where gelatin would add unwanted flavours, agar will not. In terms of longevity and storage, this is another advantage to an extent that agar has over gelatin. It doesn't really matter what form it is used in, whether that is as a flake, a powder, a strand, a solid mass, or anything else. They can all be kept the same way, and for 8 months, and in some cases longer depending on the circumstances, so long as they're stored in an airtight container, somewhere cool and away from direct sunlight or heat. The usual storage precautions. This means that you can store it for a long time and use it for a wide number of things without having to worry very much. The use of agar extends to more than just food, and we've touched on it slightly, and that is research purposes. Agar is a very powerful tool as it is the substrate of choice for trying to grow many microbes, and we have a video on that. That video will provide you details on the different kinds of agar that are used, largely owing to the modifications of what is within it, and how the agar itself presents advantages over many alternatives. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.